Okay. Can you hear me? Okay. I put the presentation on the internet. Also, I put the presentation for next week, for Thursday. I hope I didn't destroy the recording or something by doing that, but I hope it's fine. Okay. So, just for a little bit for orienting you. So we're now, this lesson will be about the cerebellum. It will be a more introduction to cerebellum, more textbook view of the cerebellum. There will be 2 hours. I'll give this lecture. And on Thursday, Yossi Yaron will give his view of the cerebellum. It will be more his view and less basic. That way you'll get also the basics of what is cerebellum and what the research of the last, I would say, 100 years, and also the more modern view or Yossi's view of the cerebellum. Let's try turning this off. Is this better? Yeah. Okay. So that's this week. The week afterwards, we have lectures of Hagai's group, the people that are supposed to talk about the basal ganglia. That's next week. Just announcement. So Hagai still has half an hour that he plans to talk. So those who are giving lectures in his session will have half an hour to talk. Okay, so it'll be half an hour Hagai, half an hour another lecture, and half an hour the last lecture. And afterwards, the next Thursday, the students that are giving about cerebellum will give the talk, and then we're almost done. Okay, so cerebellum. This is what we'll talk about. So I'll start with an introduction about a really classical neurological finding about the motor symptoms of the cerebellum. Then I'll talk about macrocircuitry of the cerebellum, what is the really gross anatomy. I understand you didn't really learn this in LSEC basic courses, right? No? You did not learn about cerebellum. Okay, so I'll talk about a little bit about the anatomy. And then I'll talk about the microstructure, the microcircuitry of the cerebellum, the classical findings about how cells are connected in the cerebellum. I'll talk a little bit and mention and the principles of the Mar-Albus model. This is like the first model of how the cerebellum works that is still relevant today. This was done in the 70s independently by Mar, that the same Mar that wrote the book about vision. I think Udi probably talked about that. And Albus was independently, they developed models of the cerebellum. Then I'll give 2 examples, maybe the best studied examples of what the cerebellum does. The cerebellum in the context of VOR, vestibular ocular reflex, and vestibular ocular reflex learning. That's maybe one of the best examples of where we know what the cerebellum does. We know a lot about what the cerebellum does. There's still a lot of unknowns, of course. And then I'll talk about the cerebellum in the context of eye blink conditioning, another example where there's a lot of information across the last, I would say, 30 or 40 years of a really nice and detailed study of how the cerebellum works and the relation between what we know anatomy and what we know about physiology and what we know about plasticity in the context of eye blink conditioning. And then in the end, maybe if I'll have some time, we'll talk about more recent research, maybe about non-motor components in the cerebellum. I'll talk a few examples of reward in the cerebellum. So let's start. So maybe the first 2 neurologists that studied or understood what the cerebellum is doing are here, Babinski and Holmes. So Babinski was a French neurologist, and maybe some of you know about the Babinski reflex. Did you hear about that? That's a reflex that if you go on the foot of a baby and you do this kind of movement, he extends his fingers. So that's the Babinski reflex. He was studying really reflexes and really simple motor behavior. And in his studies, he characterized a lot of the patients with cerebellar damage, and he found all the kind of motor symptoms that are damaged with patients with cerebellar lesion. That's one of them. And the other neurologist is Gordon Holmes. He was a British neurologist. And what he was famous is that after the World War II, there were a lot of patients with damage to the cerebellum because the helmet then didn't really cover the cerebellum. And this is what the first, I would say, massive war where they really needed the helmet. And there are a lot of veterans with lesions in the cerebellum that were shot in the back of their head. So in that sense, there were relatively clean lesions that were able, and because of that, he was able to really characterize what the cerebellum does, or what are the motor symptoms that are related to cerebellar damage. So here are 3 examples of what kind of behavior is damaged when we have lesions to the cerebellum. So the first is timing. So this is what you see here. Very simple experiment. You have a patient. He holds 2 things in his hands, and you tell him at the go signal he's supposed to squeeze both of his hands. And when you look at the hand that's ipsilateral to or contralateral to the cerebellar lesion, he does it very good. But when you look at the other hand, the hand that's ipsilateral to cerebellar lesion, you see there's this delay in activity. He doesn't initiate the movement at the right time. And then the behavior is more, I would say, not as nice, not as vigor as the other hand. So that's one example, or timing of a movement. Another example that I show a movie in a second is a simple behavior where you're supposed to extend your hand and then just touch your nose. Yes?
You show the timing one is ipsilateral?
Yeah. So generally, cerebellar damage is ipsilateral. Yeah, and I think it's written here, yeah. Left cerebellar lesion and left hand is damaged. And another example is, so you're extending your finger and you're supposed to bring it to your nose, and there are 3 types of problems here. First, the trajectory might not be straight. So if you would ask you to do it, would just simply bring your finger to your nose in a straight trajectory. But then people with lesions in the cerebellum sometimes have a more, I would say, a not straight trajectory. Another issue is, and you'll see this in a second in the movie, they have this essential tremor when they try to get to the end point. You see this tremor, and it's really hard for them to touch their nose. And another issue is essential tremor, or tremor at rest, where they have some tremor while they're holding their hand and not doing anything. And in a second you'll see a movie of this. So it's size and direction of movement, coordination, and by coordination I mean that when you need to move multiple muscles, you might have a problem with that. So the classical example is that you ask a patient to, let's say, to move his hand like this without moving his shoulders. So he's supposed to do this kind of behavior, and instead, because he can't coordinate both joints, he does this kind of movement. He moves both joints simultaneously. So when I ask you to do this kind of movement, you're actually applying force also on the shoulder, on the muscles that are related to the shoulder to stop this movement, and patients cannot do that. And another example is a pattern. So you're asking a patient to do something like this, very simple for you and most of the people, but what you can see here is when you ask a patient to do that, it's messed up. Okay, so simply patterns of activity are hard to execute. So these are 3 examples. There are many other examples. So for example, also posture. If some lesions of the cerebellum, there's problem with posture or gait. They instead of walking in like they have like a very wide gait. So there are many motor symptoms and motor-related symptoms when cerebellar damage. So here's a movie, just one example for a patient with cerebellar ataxia. Let's see. So we have damage to his left cerebellum.
Of tremor disorder, in addition to rest tremor and postural tremor, is intention action tremor seen with cerebellar disease.
So he doesn't have problem when he
in this patient who has had a traumatic hemorrhage involving one side of his cerebellar white matter pathways, he has a normal function on the right side, but the left side shows significant dysenergia and intention tremor. This type of tremor contrasts, however, with the postural tremor in that both the trajectory and the endpoint are abnormal. And most of this patient's dysfunction is in fact right at the end point when he tries to touch his nose or to bring his finger totally out. The trajectory is not normal, but the trajectory is not the area of maximal dysfunction. This type of tremor can also be seen in multiple sclerosis and other primary cerebellar disorders.
Okay. So that's just an example for, seems to see it in real life. And you can go YouTube. There are many other examples of a patient with cerebellar damage. So this is cerebellar ataxia. This is the damage to the more intentional movements, in which the cerebellum is involved. Okay. So here's just a citation from one of Holmes' patients, where he had the lesions in the right cerebellum, and he says, movement of my left arm are done subconsciously, does it simply with no problem, but I have to think out each movement of the right arm. I come to a dead stop in turning and have to think before I start again. So again, this is related to the damage to the right side is related to right movement of the right arm. But the point is that he's saying that he can do the movement. It's not paralysis of the total paralysis, but it seems like all the unconscious processing that we do when we decide, in a sense, to move our hand, we don't think about the trajectory. We don't think about what we do, but and that it seems to be part of the role of the cerebellum, just to do that calculation from our plan to move and the actual execution of the movement. If you don't have the cerebellum, you really need to think, and you really need to give all the time these motor commands of what to do at each point and really very intentionally move your arm. Another important aspects of the role of the cerebellum is learning or motor learning, and I think I showed this slide at one point in the introduction part. So this is in this case, you see this woman. She's trying to throw this arrow towards the center of the board, or whatever it is. So initially she does it very very accurately, and then you put a prism, these glasses that basically shift the view in some degrees, and then there's a difference between where you look and where you point your arm, and there's this big error in the beginning, as you see here, and then after some time you adapt to this arrow, and you throw the arrow at the right location, and then when you take off the glasses, there's an after effect, as you can see here, and then you learn it and you get to the perfect or good behavior. So this is normal people, and people with damage to the cerebellum can't adapt to that. So they have this, initially they do it well, then you put the prisms, and you can see here they make errors and they don't get better. Maybe in this example they even get worse. And then when you take off the glasses, you see it that they're back in accurate. So they didn't learn this relation, this coordination between where they're looking to how they're moving the arm, or eye-hand coordination. And there are many other examples, and I'll show later, of the kind of behavior that you require learning that are damaged in the cerebellum. Yes.
This is with late damage to the cerebellum.
This is with
Late damage, like when you're talking about damage, like
the. I'm not sure, but I guess that's the case, yes.
If you were born with...
Yeah, so Yossi will talk a little bit about people that are born without cerebellum at all and what happens there, but I think this is late damage to the cerebellum, yes. So that's learning. I'll give some examples later about what are the neural mechanisms and how the cerebellum is involved in learning, what exactly the details of the motor, of the circuitry that allows us to learn in the context of VOR, vestibular-ocular reflex, and eye blink conditioning. So I'll talk a little bit about cerebellar anatomy. I understand you had one slide, so now I'll have two slides. We're multiplying the amount of knowledge and anatomical knowledge you have about the cerebellum. So this is the cerebellum, and it's in the back of your brain here. And what you can see here are the lobes, the cerebellar cortex. That's what you see here on the outside. Okay, it continues here. They just cut it to show what's underneath. So that's the cortex. Inside here, this is deep. This is not on the surface. This is deep in the cortex. You see the cerebellar deep nuclei. See these? And this is the target of these neurons. So they project to the deep nuclei. That's the cortex. And the cortex is divided, I would say, and I'll show it later, to three main parts. You have here the hemispheres, this part, and we have here the middle line, which unlike the brain where the two sides are separate, unlike the cortex, the cortex where the two parts are separated. Here, this is gray matter. It's called the vermis, and we have another hemisphere here, and another part of the cerebellum that you can see here. This and this. This is called the vestibular cerebellum or flocculus. These and these, and are important for calculating vestibular information. And I'll talk later more about these. The cerebellum is connected to the other parts of the brain through three main pathways. You can see them here: superior, middle, and inferior peduncles. The inferior and middle are mainly inputs from the spinal cord, for example, or from the pontine nucleus. The superior is mainly an output of the cerebellum. It projects back to the thalamus and then to the brain. Another separation of the cerebellum is to the anterior parts and the posterior parts. For this, it's not. I won't talk about this anymore. But this is more related a lot of times to more like arm movements are in the interior, and posterior we have other functions. What else? Here you can see a sagittal view of the cerebellum. So we cut. This is the cut in the middle, and you can see nicely the different nice. It's called tree of life or whatever you want to call it. You see these are the. This is the cerebellum is basically folded inside in order to increase its area. And you can see each of these are called lobules, and each of them has a name. In my days, we had to learn them by heart. I guess now you don't need to learn them anymore. But then because you're not doing the neuroanatomy course, but they're different. It's a nice way to mark the cerebellum. It's, if you want orientation to know what you're talking about, then you have these names of the different lobuli. So this is the sagittal view, right? Also, when we go further away, we also have these cuts that we can see all these different lobules. And it's important to mention and show functionally what is the relation. So there are connections that go this direction into the board, and there are also connections go out this way. So functionally, there's the relation between the anatomy and the diagram. We'll talk about later. What else is important here? Well, there are a lot of important things, but I think that's it for now. In terms of the basic anatomy, when we think about input-output structure, so we take the cerebellum and we can flat it out. So we took the hemisphere that would look like this, and we just show it as a flat picture of the cerebellum. So there are three in this example. There are, I would say, again, these three parts. We have here the hemispheres, as you can see here, what's called cerebrocerebellum, connections from the cerebral cortex to the cerebellum and back to the cortex. We have the spinocerebellum, this middle part, as you can see here, and there are also sub-connections here. This gets input from the more lateral part get input from the cortex, also from the cortex, and project back and project towards the brainstem and red nucleus. And the central part gets information from the trunk and from different modalities, and also you can see here projects towards the spinal cord and the vestibular nucleus. There's a tendency of these areas to be related to more the distal parts of your arms, for example, and these parts are more related to the proximal parts and to the posture and to the trunk of the body. This part, the lateral part of the spinocerebellum. Okay, you can see this hand here. That's how this is marked. Okay, this part, and this is more to the trunk. You see, it gets information from the trunk, also related to vestibular information. That's about our general posture. And finally, the vestibular cerebellum gets the input related to from the vestibular sensor and from our eyes, and projects to, and this here, this is the flocculus, and projects then to the vestibular nucleus. Just in terms of, there's also different deep cerebellar nuclei, the dentate, the interpositus, and the vestigial, and each of them get input from different parts. So the dentate gets mainly from the hemispheres, and related to the input from the cerebellum. The interpositus gets mainly from the spinocerebellar, the lateral part, and the vestigial gets from the center. Okay, so these are the deep cerebellar nuclei, and then they project to different areas. So the cerebrocerebellum also projects back to the cortex, okay, through the thalamus. That's this pathway. This pathway, you can see here, projects to the red nucleus. I think I've talked a little bit about this pathway, right? About the connection to the red nucleus and the reticular nucleus, and also down to the spinal cord. And then this pathway, again, to the vestibular nucleus, and also to the areas in the reticular formation that project down to the spinal cord. And as I said here, the vestibular cerebellum to the vestibular nucleus in the brainstem. Yeah, that's a good point. So this is different in the sense that it doesn't project to the deep cerebellar nuclei. It projects directly to the brainstem, to the vestibular nucleus. But then if you ask Yossi Aron, for example, about this, he'll say, well, there's not a big difference. You can think of this nucleus also as a deep cerebellar nuclei. So it's not they're in different location, but in terms of function and also in terms of types of cells, there's a lot of similarities between these cells and the cells here. Okay? But this is a direct pathway, and I'll talk about later about this pathway when we'll talk about the vestibular ocular reflex learning and how to coordinate between head and eye movements. So that was the basics of the anatomy, gross anatomy, like the system. Now we'll go into the microcircuitry, and this you might have learned a little bit more. So now we're looking at a tiny piece of the cerebellum, and what we know about the cerebellum is very, very uniform. What I'm showing here, this microcircuitry, is similar in different parts of the cerebellum. This leads to the hypothesis that the cerebellum is doing the same thing but in different modalities. At this point, it's a hypothesis, we think I don't want to say hope, but we think there's this basic computation that the cerebellum is doing, but for different parts, it's doing different parts of the different kinds of behavior. It's doing the same computation, but for different things. We don't know that's actually true. And that was at least the initial hope and view. Now we know there's also differences in different areas in the cerebellum, but the basic structure is there. So what do we see here? So we're looking at a tiny piece, and we see here the layer in which the inputs, this is white matter, where all the inputs come from and how the outputs go. This is what you see here. And then this is the white matter. And then we have three layers: the granular layer that you see here, the Purkinje cell layer that you see here, and the molecular layer. The granule cell layer has the granule cells, all these tiny dots, and we like to say that, but half of the cells in the brain are at this layer, and the cerebellum is. If we look at the total number of cells in the brain, about half are outside of the cerebellum, or half in the cerebellum, and this tells us something about how much processing is happening there in the cerebellum. So we have this granule cell, have these tiny cells. They get input from their input from the mossy fibers. So we see here's the mossy fiber. This red fiber it terminates here, and it connects to the granule cells. Okay, so that's what's happening here. After that, the granule cells project what is called the parallel fibers. These are the granule cells. They send these axons to the molecular layer. The name of?
The granule cells project to?
The granule, they send the parallel fibers. Their axon is called the parallel fiber. This is this. And the reason it's called the parallel fiber because it goes up, and then it sends these parallel fibers across the cerebellum that you'll see here, and I'll talk about this more. And the molecular layer is basically where all the dendrites of the Purkinje cells are, and all these parallel fibers are. So we have these parallel fibers in the molecular layer, and here we have these huge dendrites of the Purkinje cells, and these parallel fibers connect to the Purkinje cell. That's what you see here. And then the Purkinje cell layer is simply where the cell cell body of the Purkinje cell is. It's a layer. You can think of it as a two-dimensional layer that covers all the cerebellum with these Purkinje cells. So that's the basic unit of the cerebellum. And let's talk about this with a little bit more detail. And I forgot, of course, the very important climbing fiber. That's what you see here. These are fibers that come from the inferior olive, and I'll talk a lot about them, and Yossi will talk about them a lot more. These are fibers that come from the inferior olive, and they connect very, very strongly to a Purkinje cell. That's what you see here. So you see this goes here, really connects very, very strongly. One fiber has thousands of connections with a Purkinje cell, a very reliable connection between the two.
Where does the muscle fibers?
So the muscle fibers come from many places, but mainly from the pontine nuclei. And the pontine nuclei I didn't mention before, but they get information from all around. They get from the connection from, for example, the cortex to the cerebellum goes through the pontine nuclei that connect to from them come all the muscle fibers. Yes.
Just to make sure these nuclei are in the cerebellum or
So this is the cerebellar cortex. Okay, it's all right. This is all the output of the cerebellum. It's the cortex. This is how it looks. When you say nuclei, which
you talked about where the climbing fibers come?
they're in the midbrain. They're somewhere here. And the pontine nuclei are in the pontine here.
The climbing fibers make other synapses only with one Purkinje cell?
So I'll talk about it later. But basically, each Purkinje cell gets one climbing fiber, but one climbing fiber can project to a few or up to 10 Purkinje cells. So it's but what's important, each Purkinje cell gets only one. And again, this is something that happens during development. In the beginning, it doesn't look like this. There could be few connections, and during development, in the end, we have a single Purkinje cell. Purkinje cell gets a single input from one climbing fiber. It's similar, for example, in our muscles. And during development, we have the same thing. Each muscle fiber initially gets a lot of connection from many, many neurons. In the end, after development, you're left with one neuron that connects to a single muscle fiber. So this is, and I'll go more deeply into the details of this circuitry. So the first, of course, we start every neuroscience lecture has to have Ramon Cajal in it. So he was the first to really describe these cells, the Purkinje cells. And here you can see his nice drawing. You see the cell body of the Purkinje cell, and you see the huge tree that these cells have. Very nice. And this is the basic, I would say, direct pathways from input to output in the cerebellar cortex. So what do we see here? Here you see what comes here, from the fibers to here. Which fiber is this? Muscle fiber, exactly. And they connect to the granule cells. You see them here. They have a very special connection that's called the glomeruli. I'll talk about this later. Each granule cell here has about 4 connections. Number 4 is interesting. There's a recent paper of Larry Abbott that says, well, maybe in some sense this is actually optimal in this circuitry. And I encourage you to go and read that paper. It's a nice paper. So they connect. Each of these granule cells have 4 connections. They connect to 4 muscle fibers, and then they arise this fiber, this pathway, the parallel fibers, or at this point they're still not parallel. They go up, and then they go parallel here, and connect to the Purkinje cells. That's what you see here. So the Purkinje cell, what you're seeing here is that when we look at, imagine this is the cerebellum, this is front. You're seeing the Purkinje cells at this orientation, right? And you see they're very, very thin. If we turn it at 90 degrees, you'll see something that looks like this. So they're very, very thin, but they have a huge, huge dendritic field. Okay, is that clear? So what you're seeing here and here is the same thing, but this is flipped to 90 degrees like this. And what you can see here is when we look at the parallel fibers, each parallel fiber doesn't have an opportunity to send to connect very strongly with this with the Purkinje cell because it's very, very thin in this direction, And indeed, if we look at the amount of connectivity that we see here, there may be 1 or 2 synapses from each parallel fiber to each Purkinje cell, but then the parallel fibers pass through many, many, many neurons. And you see this is very organized, and if you compare it to other circuits, very, very organized circuit with very characteristic connectivities. And that really led to a lot of hypotheses of how we think the cerebellar works. And maybe one of the reasons that we understand a lot about the circuit is because of this order and the way which we can look at it and identify these specific connections. So that was one pathway, the pathway from mossy fibers, granule cells, parallel fibers, Purkinje cells, and then output. And the output here is to the deep cerebellar nuclei or vestibular nuclei. That's what you see here. And the second pathway that I talked before, these are the climbing fibers, and you see them here. Climbing fiber connecting very, very strongly to a Purkinje cell. So this is still only anatomy. We're getting close to physiology pretty soon. And this is just a caricature that's showing the same. It's the same thing. I'll use this more because it's easier to see, and there's a lot of colors, and you can identify the different elements: the mossy fibers, granule cells, Purkinje cells. So we talked about this. And then just in terms of number, so parallel fibers, we have about 10,000 to 100,000 for one Purkinje cell. Okay, each of them could have a few connections, not many connections. In terms of climbing fibers in this pathway, we have one climbing fiber can connect to between 1 and 10 Purkinje cells, but each Purkinje cell gets a climbing fiber for only one cell. Gets only one climbing fiber. So this is what you see here. Okay. So then that's only part of the story where I have a little bit more, a few more cells. So these were the direct connection from input to output. We also have interneurons in the cerebellum, 3 main interneurons. The first one is the Golgi cell, you see here. It's in the, and I'm mentioning this, and I'll talk about this a little bit more later when we talk about the Eibling conditioning, the function in Eibling conditioning. But its cell body is it's in the granule layer, as you can see here. So it gets input, the same input that the granule cells get. Okay, you see it here. This is where also all the granule cells get their input. So it gets its input from the mossy fibers. Okay, and it also projects to these. It also projects to these. Sorry, mistake. This is the axon of the cell. So what I wanted to say that this is connected. This can connect to the granule cells and inhibit them. They get input in this layer, in the molecular layer, and then there's this feedback loop where you have input from the parallel fibers that get here, and then it activates this cell, and then this cell can inhibit the same granule cell that excited. So they have this negative feedback loop between the Golgi cells and the granule cells. So it gets here. This is the axon. This is the dendrite. So from the parallel fibers in the molecular layer. So they have these. We have the granule cells. They go up. They also give information to this cell here. So now we know that's not true, but that was the initial thought. Now we know they also have collaterals that they can inhibit other cells, but mainly to the deep cerebellar nuclei. But this is the interneuron again in the cerebellar cortex.
Just like they do and get inputs from the
Here, I'll check that. For sure they get input from the parallel fibers, and I think this is the axon where it connects to many granule cells. But I'll check that. So that's one interneuron, the granule cells, and I said there's the negative feedback between the input that it gets from the parallel fibers and back. It inhibits the granule cells that can also excite it. We have another 2 types of interneurons. The satellite cells. These are interneurons that are in the molecular layer, and they also their axon is also in the molecular layer. And what they do is inhibit the top part of the molecular layer, or the distal tree of the Purkinje cell. So they can inhibit this part of the Purkinje cell here. That's one kind of interneuron. And the other kind is the basket cells that you can see here. They're closer to the cell body of the Purkinje cell, and they also have these strong inhibitory arborization on the Purkinje cells themselves. That's what you see here, here, and here. So these are more inhibiting the dendrites of the Purkinje cells, and these are inhibiting the cell bodies, or closer to the cell bodies of the Purkinje cell. The basket, yeah. As you can see, see them. These are here, and these are their axons. Imagine you have a Purkinje cell here. They're called basket, but it looks like a basket that sits on the Purkinje cell. You would think that this is totally, and I did mention this is very important. All of these three interneurons are inhibitory. Okay, so they reduce the probability that the presynaptic cell will fire. You would think that this basket simply silences this Purkinje cell, so we would think that when this fire, you'll have a complete inhibition, for example, of this cell, but that's, when you look physiologically, you see that's not the case. There is a connection, it is inhibiting, but it's not completely silencing these cells.
Is there any theory of affecting the calming fibers, or what's the benefit of having many synapses on dendrite and not a really big synapse?
Yeah, the thought is that when a climbing fiber fires, it causes a huge, that's known, it causes a huge calcium spike in the Purkinje cell. And the theory, or one of the theories, and Yossi will give a different theory, as this is an error signal, a signal that tells you to learn. And in that sense, it makes sense to make this, to connect to all the dendrites, because you want to learn across all the dendrites. So that's the thought about this, but we'll get to that later. And there are other interneurons that they're not. This is the canonical diagram where you see across all the cerebellum. There are other interneurons that are distributed differently in different areas. One important one, I think, maybe for Noga, your talk, is the unipolar brush cell, which is an excitatory interneuron. I'm just mentioning it. I won't talk about these anymore. There's also neuromodulators in the cortex. For example, serotonin is really important for cerebellar processing. So almost finished with the anatomy. There's one other structure, one important structure, the glomeruli. So basically what you see here, this is this small part, is the connection. You have here the mossy fiber that connects to the granule cell. That's what you see here. And you have the Golgi cell inhibiting this same synapse. And they're packed in this structure that you can see here. So we have the mossy fiber giving input to the granule cell neurons, and you see them here, several of them. And we have here the Golgi cell that sits on the same structure and can inhibit it. Okay, so that's where the feedback from the Golgi cell can influence the input of the molecular layer. In some numbers, so we have one mossy fiber can connect to 20, around 20. These are all estimations. Around 20 of these. So we have one mossy fiber can split to 20 of these glomeruli. The glomeruli itself gets input from one mossy fiber. It gets also this input from this Golgi cell, this inhibition. In terms of output, we can have 50 granule cells connecting in this point. So if you do the math, we have one mossy fiber connecting to 20 glomeruli, which then connect to 50 granule cells. So this is an order of 100 increase from one fiber to 100 granule cells. 1,000 granule cells. But remember, the granule cells have like 4 synapses, so it have 4 of these. The granule cells connected to 4 of these. So we have about one mossy fiber connecting to 250, leading to an expansion from one mossy fiber to 250 granule cells. Or again, these are estimation. If it's 100 or 300, for me, currently it's the same. And we said each granule cell has 4 dendrites connecting to 4 of the glomeruli. In the end, the Purkinje cell takes this, and there are around 200,000 connections here to each Purkinje cell. Another anatomical organization that, and this is the last one, and I don't think we'll talk about this functioning. Maybe Yossi will return to this. But there's an organization between the cortex, the cerebellar cortex, and the inferior olive. Okay, there's a specific pattern of projections. So if we look at different areas of the inferior olive, as you can see here, and they're in different color, they project to different areas in the cerebellum. So this part of the inferior olive, this brownish part, will connect here. Then this yellow will connect here, and it makes this stripe, right? This a stripe organization of the cerebellum based on its input from the inferior olive. And it's interesting because you think of these, we have all these Purkinje cells here that are organized like this, and then we have the parallel fibers that are going in parallel to this organization. And it's a way maybe to converge input from two sources. One from the inferior olive that has this kind, and then you can modulate, as you can see here, the input here based on different, I would say, horizontal, sagittal stripes. That's what you see here. So we have here the mossy fibers connecting, and they're going this direction, and then the connection from the inferior olive is connected this direction. Okay? So we have two types of patterns of connectivity that they converge, and the question is, how does this lead to the function? And then these stripes also project to the same area in the deep cerebellar nuclei, and that's what you see here. And that we know. So the more lateral areas, for example, would project to the dentine nuclei, and then when we go, as we go more and more medial, we project to different parts of the deep cerebellar nuclei. So that's another organization of the cerebellar cortex. Yes?
Yes, I don't understand the olive nuclei. I don't know. What is the organization there?
So that.
What happens there?
So there are different areas here in the inferior olive that connect to different areas in the cerebellar cortex.
Yes, but is there a military organization in inferior olive?
So they get input from different. We know less about the exact inputs to the inferior olive from different areas of the brain, but they do get slightly different input. So the cells that, if you have an inferior olive cell that fires here, it's not. It won't fire to the exact inputs here. No, they could be also sensory inputs. So, for example, from the visual inputs, there could be input from the spinal cord about different kinds of receptors, but all kinds of inputs. Okay. So, in the end, what we have after all these the detailed anatomy, what we have is like the canonical diagram of the circuit. So we can think of, and we'll return to this when we'll talk about the function of the cerebellum. So we have here the mossy fibers connecting to the granule cells, to the parallel fibers. We have here they still have one interneuron. We mostly ignore these interneurons, and they project to the Purkinje cells. Purkinje cells project back to the deep cerebellar nuclei. That's what you see here. So this is the pathway from mossy fibers back to the deep cerebellar nuclei. And this is, and importantly, the Purkinje cell is inhibitory. I didn't mention that, but that's important. So when this cell fires, it actually inhibits the cerebellar output. This is the deep cerebellar nuclei. But in many cases, there's also a direct pathway from input to the deep cerebellar nuclei. So you can think of this as a direct pathway for a motor command, for example, which on top of that has this control pathway that goes through the cerebellum. Okay, so you can give a command and then modulate it through the activity of the cerebellum and use all the learning abilities of the cerebellum to modulate that. So that's this. And of course, there's the other pathway, the inferior olive pathway, that sends the climbing fiber to the Purkinje cell. And in many cases, the inferior olive, the input it gets from sensory signals. So I talked about sensory receptors on the skin. For some parts of the cerebellum, in other parts, the sensory signal could be, for example, information about visual motion. And that's in the context of VOR, I'll talk about that. So this is sensory information. And the idea now is that what the cerebellum can do is get this, the brain can send this command. Now we can modulate it through this pathway based on that sensory information that we get. And this kind of structure is thought to be important for correcting errors. So think you're doing some kind of movement, then you get some sensory signal that this movement was done incorrectly. Then you can modify this connectivity and then affect the way this direct input is from input to output to motor output is modulated. So that's the basic idea of the circuitry. There's another connection that I did not mention. Again, Yossi will talk about this more. There's a connection from the deep cerebellar nuclei to the inferior olive, which you can modulate it. And it will be also relevant to one of the students' talks in the end. So this is the anatomy. Now, luckily, we don't have only anatomy. We also have we can also look at the physiology of these structures and look at the physiology of different cells in the cerebellum. So from the 60s and later, in the 60s it was done mainly in slices, and later they did also in behaving animals. We're able to record activity of the Purkinje cells. So we're now recording activity in the Purkinje cell, either intracellularly or extracellularly. That's what you see here. And what the researchers noticed, there are two types of spikes in the Purkinje cell. So in this case, we're sticking an electrode into the Purkinje cell, and we're recording its activity. And this is what we see. We see these spikes here, and these are called simple spikes. And these simple spikes, you can increase their rate if you, for example, stimulate the parallel fibers or the MOSI fibers. If you stimulate here, you'll get more input from the granule cells, and then these cells will get more excitatory input, and then you'll get an increase in these spikes. This is what you see here. But then when there's a second type of spikes, the complex spikes, and that's what you see here. And the word simple is because this seems simple, and complex because this is complex. This looks more complicated. And it turns out that this kind of spike, this very messy spike, happens when you activate the climbing fibers. So the idea here is now that you're activating a single fiber that's connected many, many, many times to this neuron. This spike opens many channels and causes this very complicated spike. And it turns out that these spikes, the simple spikes, are regular sodium spikes that happen that happen in the soma, whereas these complex spikes are actually calcium spikes. It's a huge influx of calcium into the cell. And we know from many different studies, from the hippocampus to wherever you want, that calcium, that entrance of calcium to a cell is many times related to learning. And I'll show later how, and I'll show later that indeed when a complex spike fires, there's changes in the synaptic weights across the Purkinje cell. So this is intracellularly, which we can do mainly in slices, but what is nice, we can also identify these cells also extracellularly. So we put an electrode close to a Purkinje cell, and we record the activity. So now this is the electrical field that's measured around the cell, and we can see these spikes that are happening all the time. And then it's hard to see here, but here there's a complex spike at this point, and then many more simple spikes. So when you look more carefully and you increase that scale, you can look at the simple spike, and you can look at them and see them here. And this is an experiment where they stimulated the MOSI fibers and recorded activity. So you see here, this is the point where they stimulated, and you see there's this simple spike. So they did this, I think, twice in this case. So you have here one simple spike, and the next trial you have another simple spike. Okay? And this is just with extracellular recording. And we can identify these kinds of spikes. But we can also identify complex spikes. So here you give a stimulation in the climbing fibers, and this is what you get. You get this kind of pattern where you have one big spike and then many other spikelets. And these spikelets are actually what's happening at this point.
What makes this one complex spike?
In what sense is it one? That's a great question. And indeed, it's one because we stimulated one climbing fibers. In that sense, it's one. You're asking, I think what you're asking, well, what happens here in the axon of the cell? What is it telling the world, right? And it turns out there's not a clear direction. It's not that each of these small spikelets is actually a spike going in the axon. Some of them are actually spikes that are happening in the dendrite itself, and not all this is going to the axon of the cell. There's usually a spike. When the complex spike happens, the axon sends one spike usually in the beginning, and maybe another spike in the end of the complex spike related to this last spikelet.
We measure this from the...
This we measure from close to the cell body or around next to the cell dendrites. So what happens in practice, a lot of times what happens, if you remember the organization, if you go here with an electrode, extracellular electrode, you get close to the cell. Many times when you're in this layer, okay, even before you actually see the simple spike, you can hear already the complex spike because it's actually all the axon, all the dendrites being activated. So you put an electrode in the beginning, get only complex spikes. Then when you get to the cell body, you start to see these simple spikes.
So this is even more convincing that it's not one spike because it's from different areas.
Different dendrites. Yeah. It's different. It's spikes that probably each spikelet could be from a different dendrite, for example, or a different branch of the dendrite.
But it's still considered one.
One, yeah. It's one in the sense, I said, one in the sense that it's activated by one climbing fiber. And one important question, then this is another relatively recent Nature paper in which they looked at is there any information that's given in the length of these complex spikes? So we can have, when we record, sometimes we see short complex spikes, like you have only one spikelet. Sometimes we have two, sometimes we have four, or whatever. And it's different for different cells. It also depends on where exactly you are relative to the cell. But what was nice, they were able to link between the size of the complex spike that you record extracellularly to the actual how much learning the animal did. So in a sense, big complex spikes are related to more learning. Yes?
Make sure the multi fibers give the single spikes and the
Climbing fibers, yeah.
And when you say size, you mean the length of the
Yeah.
fibers?
Yeah. What is nice with extracellular, now you can go to animals and wild animals' behavior, look at the signals. Another, I think, why this made a lot of this lead to a lot of important experiments is that we have here a condition, and this is rare, where we record a single cell, but we can actually dissociate between two kinds of inputs. And not just arbitrary two kinds of input, one that we think is related to a signal to learn, and the other to the signal of how much to excite the cell. So the convergence of both of these inputs led to a lot of important experiments that led us to understand or get as close as possible I think maybe closest than any other area, brain area, to how this system learns. Let's see, how much time do we have? Okay, maybe I'll do this and then we'll go to a break. So we talked about that, and we saw in the neurology there was a lot of relation to motor performance and to motor learning, and this was already understood in the 60s and in the beginning of the 70s, and it would show what basically at this point there was very little evidence about actual behavior in cerebellum in behaving animals. There was mainly anatomical information. And based on this anatomy, mainly Marr and Albus came with these models of the function of the cerebellum. And their idea is that the cerebellum is important for motor learning. So here is an example for motor learning. This child learning to ride a bicycle. In fact, there's also another motor learning, the father trying to teach the daughter, but that's a different story. I'm not sure what's harder. Anyway, so this is the context of motor learning, and there are different kinds of motor learning. I'll talk today about vestibular ocular reflex, or learning to adapt to changes in the gain of vestibular ocular reflex. There's also learning to adapt to. We talked about prisms, learning to adapt to loads of arm movements. There are many different contexts that we call motor learning, and and this is from Albus's paper. What they suggested is that the way the cerebellum works is basically similar to a perceptron. And you learned in Neural Networks 1, right? No. You did not learn about perceptrons? Oh. Okay. So the idea here is that you have this set of inputs that you see here, and we have these weights, W0 until WM, whatever, how much input it has. And then all these inputs converge to a single cell, so you can think of this as the cell body. The cell body sums all these inputs, these weighted inputs, okay? And then based on this sum, with some threshold, decides if to fire or not to fire. And what a perceptron can do, a simple one, is basically characterized between things. Maybe do we have here? So the basic idea is that some of you probably learned this, right? This will work. Is there any? The basic idea is that we have, let's say, this is input 1. Let's call this x0, and this is input 2. This is input 0, this is input 1. And now we want to characterize something. So let's say here you have something that its output. This is the output. This is plus, plus, plus. And here you have a minus, 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 minus. I don't know, minus here, minus here. What the perceptron will learn is to calculate the line that separates between this and this. So this could be pluses. I don't know. You can have here, I don't know, things that are on 2 legs versus things that are on 4 legs, whatever. You can characterize between different things, but the idea that's the basic idea. In terms of motor control, we can think about inputs that we have that we want to generate a specific motor command based on inputs. So that's the idea of the perceptron, and this reminded at least Mar and Albus of the organization that we have in the cerebellum. So we have, ignore this at this point. So we have all these inputs that come to the cerebellum. And we have this Purkinje cell that gets all these inputs from many, many, many cells. And then we have this one output from the Purkinje cell. And another important thing is the perceptron, that it can learn. And the way it learns is that it looks at it sends its output, and then it compares the output to the actual output that we have in the world. So for example, I'm trying to characterize this as a plus. I have some kind of set of weight before the learning, and then after, and then I compare. I run to the perceptron. I give the perceptron the inputs. The inputs are multiplied by this w, and then sum, and then I get the output. And I ask, is this output indeed a plus? Is this a 1 or a 0, for example? Yes.
Can you give an example of what you're trying to classify?
So I said, let's say you're trying to classify a function. This is the actual function. You have a pluses here and minuses here, and you want to separate between the plus and the minuses.
Yeah, that's like a concrete example.
So a concrete that I tried to draw that didn't work. Let's say you try to separate a visual. I don't know. You try to separate between things that walk on 2 and things that walk on 4. You get some visual input. So in the motor, let's say you get input about, and I'll talk about more in the context of Arbling conditioning. You get an input about, let's say, a tone, right? And you want to know what to do with that tone. If you get an air puff afterwards, you learn that to blink before. So the tone, in a sense, is the context or no tone, and what you're trying to learn here is a set of weights that will make you blink in the end. That's the basic idea. Again, this was done. These models came before we actually had the exact motor essays that I'll show later. And one of the criticism about these models, okay, this is nice for classifying. How exactly is this related to this? And that's what you basically ask, and that's a great question. And the basic idea of classification, I don't think anybody really takes that too seriously, classifying. But the idea that we have here a system where we have inputs and we can learn on it, I think that's something that's still influential until today. Let's just finish this analogy. So we're giving these inputs between some kind of inputs. We convolve them or we multiply them by the weight and we sum them, and then we get the output, and we can compare that output to the actual output that we're trying to do. And we can ask, and that's the error signal. And their idea was that this error signal comes from the climbing fiber. So the climbing fiber, which is the sensory input, basically tells you, did you classify correctly? Did you do, or in the context of behavior, did you do the right behavior? So that's the climbing fibers. And one point that they made, or especially Albus, is what is the advantage that we have here? So we have, just to remind you, I talked here about this connectivity. We also have these mossy fibers, and we have this expansion from one mossy fiber to many, many, many granule cells. And the idea is that this part allows us to expand the input of the mossy fibers. Instead of having this limited connectivity, this limited linear connectivity, now if we expand the inputs, we can dissociate between much more patterns. And I'll give you just the intuition, not the actual details. So you can imagine there are patterns that you can't separate with a simple linear decoder. So think about the weight as the line that separates between things. So imagine you have a pattern like this in the world that looks like a XOR. Sorry, these are minus. So there's no line that will separate the pluses and minuses to different regimes in this graph. But now if we do simple mapping of which, for example, we have a x1, x0, and x1, we can now map them together before we get an input, or we can map them. We can have different maps, and this is this intermediate layer where we expand their coding, and now we can generate when we expand them and we get these inputs, instead of using a simple direction from x0 and x1 to the output, we can expand it to many, many different intermediate computation. And now if we do this, we can actually separate. We can actually find something that will separate the two groups. And I know I'm not giving all the details, but that's the idea of this part. It will expand the coding of the simple mossy fibers, and that will increase the amount of patterns that we can separate with the cerebellum.
How does this relate to the anatomy?
Yeah, so it relates to anatomy here. We have one mossy fiber that connects to many, 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 many granule cells. And then there's this expansion here. They wrote 1 to 100. It could be even more. So we have this input that expands, now connects to many, many cells. That's what you see here. And then they converge back. And their idea that this expansion will allow to increase the amount of patterns that we can separate by the Purkinje cell.
Yeah, but here is also 3 layers, right? So, but I don't know if I understand correctly. So, mossy fiber to the parallel fiber to the clamping fiber. So, it's 3 layers conceptual, right?
There's the climbing fiber. That's this input. That's not related. It's there's this is, if you want, this is the input layer. Yeah. This is the intermediate layer, and this is the output layer. So that's 3.
Yeah. So it's 3 layers.
Yeah.
Right?
1 input, 2 intermediate, 3 output. Okay. So that's the idea, and I think it's a time for a break. Serve 19, 10:19. Yeah.
So only 3 layers is enough for doing this complex
So that's excellent question, and that was their initial idea. Now there's one thing that we're ignoring here is all the interneurons, right?
Yeah.
And the interneurons have also feedback loops in it.
Okay.
So it could. So the other theories, maybe I'll turn it off. Other theories of the cerebellum say, well, there's nothing. There's the feed-forward perceptron. There's also these feedback loops that allow you to force. Okay.
And actually, from Dan's talk, he 1 neuron. It's responsible for 7 layers of the.
Yeah, that's a possibility.
Yeah. 7 layers of the perception.
Yeah.
So probably here have maybe
Multiple, yeah.
multiple layers.
This is really, this is that. When you think about this, is I don't know, 10 years after they made up that perceptron, right? And then that was their first attempt to apply it, right? And it seemed to them natural to apply to this kind of system, right? Where you have all these inputs and outputs. But I agree that, there could be tons of other things. But this simple idea is still influential in the sense that we think of these signals as modulating the input, right? This current signal modulating the input, allowing to do something, or for example, motor control. So that is still influential. I agree that we can add a lot of levels of complexity here, and you have even a single neuron can have multiple perceptron and things like that. Although there is only I don't know, can a single neuron also diverge information?
Diverge?
Yeah. So here we have divergence of information, right? So one multi-fiber connects to many granule cells, and this leads to divergence and to expansion of the ability to cope. And what is the equivalent in a dendrite? In a single dendrite, I guess you can have, because you have many dendrites, you can have many multi-fibers connecting. Yeah, I think you can get it. Understand what I'm missing? A single nerve? Yeah, I think that's it.
בגדול, לבדוק דברים שהם high frequency, אז אי אפשר, במובן של שיש שני אינפוטים נורא דומים, אבל האוטפוט מאוד דומה. Okay, you can ask like what type of problems like you can do just with a single layer nonlinear or one layer.
לא מלמדים אתכם פרספטרון בשיטות אחד, לא מלמדים אתכם? בסופו של דבר.
I think they also teach it in
גם לא לימד?
information processing.
נראה לי אולי בסמסטר הבא. זה דווקא לא קשור ללמידה בסמסטר הבא.
בעידוד מידע ולמידה אולי.
I think they're learning both.
נראה לי גם הרשתות של סמסטר הבא אמור להיות קשור ללמידה.
כן, כל זה גם בעיבוד מידע ולמידה.
זה פיזיקה בטוח לומדים. לא?
כאילו, אני למדתי את זה כאילו סתם לפני נובמבר 2009, כאילו בפיזיקה. כאילו.
חשבתי שאתם יודעים את זה טוב. מה זה? למדתם את זה בקוגניציה. כל ה...
למה כשאמרת דיברת על האינטריורים יותר הרבה לא מספרת על למעלה ולמטה?
לא זוכר. לא בכוונה.
זה לא, אותם תיקונים, נכון? כאילו שיעור לא משתנה שם מבחינת ה...
אנטיריו, פוסטיריו...
כאילו...
איזה...
לא, ככה וככה.
אני לא זוכר שאמרתי אנטיריו, פוסטיריו. כאילו, את מתכוונת... כן, אנטיריורי הייתי חושב שזה מכיוון החוצה של קורטקס. או... לא, נכון. אנטיריורי, אבל מקופל גם.
אנטיריורי זה שם, זה רגיל, לא? זה לא יהיה למעלה. כאילו...
אני מנסה לחשוב, אם אמרתי אנטיריורי פוסטיריו, אני מצטער, כי זה לא נראה לי כיוון... כן, אז זה יהיה באמת החוצה, אבל אין פונקציונלית, אני לא מנסה לחשוב על זה משהו חשוב.
כי אמרת, אני לא זוכרת באיזה ציר זה היה, אבל באת
נסתכל בסרט. יכול להיות שהתכוונתי להגיד באידיולציה להבין אותה.
לא מאידיאלציה, זה כאילו בין דורסל לוונטרל לסטריאו.
יכול להיות שסתם כי ככה זה היה בשקט, לא בכוונת... פה?
עוד?
נגמר.
כאילו...
לא משנה.
אם התפנים משמרים אז סבבה.
מה? זה מאחורה, בחיות שונות יש להם זוויות שונות כאלה, אז מי שעל 4 או הזה, אז הסרבלום הוא למעלה, אצלנו הוא למטה דווקא שלנו, הוא כאן. למשל בעכברים מאוד קל לרשום יחסית בסרבלום, או בחלקים של הסרבלום. אפשר גם לעשות אימג'ינג בסרבלום, כי מחבר יושב ככה, הסרבלום נמצא פה, מאוד קל לא צריך לעבור דרך הקורטקס. בקופים או בני אדם, ונמצא פה, אז הגישה היא מאוד קשה. אנחנו נשארים בו. יותר קשה. ומה זה דבר לא נראה לי שהם חושבים? מה לא לינארי? לא, אז את יכולה לקחת...
אז אם זה היה...
כן, כי כאילו... יש לך בעיה לינארית כן,
כן.
כל הסוגים השונים של הנוירונים וכאילו איך שאנחנו לוקחים, כמה זה כאילו סוג שונה של תא שכאילו אתה יכול להגיד מהחלוקה וזה, אז כמה זה כאילו לפי...
אז יש חלק שכן. מה שלא כל כך יודעים שהוא ברור שהוא לגמרי שונה, זה הסטטלייט והבסקטל, אלה שנמצאים באזור של הדנדריטים, פשוט נמצאים באזורים שונים, זה לא איזושהי חלוקה חדה ביניהם.
הם מורפולוגית או רק?
הם שונים קצת מורפולוגית, אבל לא ברור שזה פשוט לא הבדל במיקום שלהם. כל השאר, פוקינג'יס זה שונה מגרנמלסר.
כן, טוב, כאלה שהם אינביטורים וכאלה ממש...
אז באינביטורי יש שם איזשהו זה, הגולג'יסל הוא גם נמצא במקום אחר, ונראה אחרת, הוא גם אחר. אני לא יודע בדיוק בהתפתחות מתי יש את הדיפרנציאציה של כל הדברים האלה, אבל הסרבלום, אחד הדברים שכן יכולים בהתפתחות בסרבלום, שהרבה דברים שקורים בהתפתחות קורים אחרי שאתה נולד. אז הרבה מהמדידה של התאים שם קורית רק בזמן ההתפתחות. וזה גם אחד התחשוב, זה תינוק באמת, הוא מוגבל מאוד במוטוריקה שלה, וככל שהוא גדל.
אבל בטח בבני אדם אולי, אבל כאילו זה...
בחיות אני לא.
איך תגיד לך על דברים כאלה בבני אדם?
הסטודנטים...
נראה לי הרבה כאלה
הסטודנטים, מה שאתה אומר כרגע זה כאילו ספציפית בבני אדם, זה מאוד שונה בקופים.
לא, מאוד דומה. מאוד דומה מבחינת הסוגים השונים של הסטודנטים. הסרבלום יחסית דומה בין חיות שונות. רק אני אומר שההתפתחות, הזמן של ההתפתחות הוא טיפה שונה, הוא מאוחר בבני אדם, ביחס נגיד לקוף. או חלקים שלו. אבל אני אומר, מה שאני לא יודע זה את כל הדיפרנציאציה, מתי קורה בדיוק מה ואיזה תא נודד לאן ואיזה קונטקסט זה אני לא.
היה... לא מזמן בוויינט? עם מישהי ששיחה, אבל זה לא היה.
מחקר פורסם בוויינט.
מוכרח שאין מוח
מישהי שנולדה בלי סרבלום.
כן, כן, יש יופי דבר. התחלה... איכשהו חיים טוב, כן, זה עד יש גם דוגמאות לאנשים שחיו, נולדו בלי קורטקס, וגם כן איכשהו חיים.
לא, זה מישהי מבן גוריון, חסנה חפות של FMRI, היא עשתה מחקר על שפות, והיה איזה בחור שדיבר על הרבה שפות, וזה ממש נגנבה, ואז היא לקחה אותו ל-MRI, ופשוט לא היה לו
יותר טוב.
בלי כופות.
כן, אתה יכול ללמוד אותו. כן, בשפות ב' אני חושב שיש עניין של השפה הראשונה שאתה לומד, שמשפיעה על שאתה לומד שפות אחרות, איכותית שונות.
לא יודעת אם זה קשור, כאילו, יכול להיות ש...
כן, מעניין.
אבל פשוט היה לו ממש מגניב כזה, אם אתה נבדק ממש שווה.
עכשיו שצריכה לשחזר את זה.
כן.
שבוע הבא באיזה יום?
ביום ראשון, אני מגיעה למכתב.
כן, את לא מציגה, אמרת היית מוכנה לעשות עבודה.
כן, כן, אבל ילדים בהתחלה,
לא. החומר כן, רק משהו לא. אני יודע, אבל תגיעו לשיעורים שלנו. Continue. Right. Quite good. So I already see that we won't get to the end of the lecture as I hoped, but we'll see what we'll do with that in the end. Okay, so now I want to start to go into the actual learning mechanism and learning an example for where the cerebellum was actually studied in the context of learning. And first, so the two main points that, or the main point that was made by these models is that we have here this input that comes from the parallel fiber, and that input could be modified by error. And this error comes from the climbing fiber. And the idea here is that, and it was splitted by Marr and Albus. Marr thought that this error could actually increase the strength of the connectivity from the parallel fibers to the Purkinje cell, and Albus suggested that will decrease the strength, and Albus was right. When a climbing fiber is activated, the strength of the connection between the parallel fiber and the Purkinje cell is reduced. And the evidence, the most clear evidence that came from... There will be no evidence that I said it. So I can say whatever I want. Now, so we have here these parallel fibers, these input to the Purkinje cell, and we have this climbing fiber. So when a climbing fiber fires, and it causes this huge complex spike and calcium signal, that will reduce the strength of connectivity between the parallel fiber and the Purkinje cell. So the evidence for this came from a work of Ito in 1982, and the kind of experiments that they done are illustrated here. So they record activity in intracellular activity in the Purkinje cell, and now they activate the climbing fiber, and they do this in two conditions. They have two different parallel fibers, so they're recording here, they're activating the climbing fibers, and they're activating two types of parallel fibers. Okay, and now they're either, let's say, they're activating this cell together with the climbing fiber, and this cell they're activating without the climbing fiber, this parallel fiber. Okay, so that's so they have two parallel fibers. Okay, parallel fiber 1 and parallel fiber 2. In the beginning, they just activate both of them. That's what you see here, just as a training. Then parallel fiber 1, they activate it with the complex spike. So they do this and this together. That's what you see here, this yellow line together with the black line. And parallel fiber 2, they activate it without the climbing fiber. So, and what the hypothesis, if this is really related to reduction of a parallel fiber connectivity, the strength of parallel fiber connectivity to the Purkinje cell, you'd expect that if you do this protocol, only this parallel fiber will be the strength of this parallel fiber will be reduced, but not this. And this is exactly what they found. Okay, and this is what you see here. So when parallel fiber 1 was paired with the complex spike, so this is before training. This is the size of the EPSP before training, and this is the size after training. So we see this reduction in the size of the EPSP in parallel fiber 1, but not in parallel fiber 2. So that's the idea, and this is from their paper, from Ito's paper, where they have some kind of test before, then they do this stimulation of parallel fibers and complex spike, and afterwards they see this reduction. Interestingly here, they see this continuous reduction, which you don't really know what's going on here, but generally there's this reduction in the strength. So this is an example for a LTD, long-term depression of this synapse based on the climbing fiber activity. And that was and again, this is something that it was hypothesized by the theories, and then they actually found it in slices. And now, so we did one level of trying to use theory was done. They had some kind of prediction about how synapses might change. But now we want to take this into behavior and ask, well, we have this simple mechanism that can induce learning. How is that actually related in the context where an animal or a human is actually really behaving? So this is the first example, and I'll just show this as an example. And this was the first example that showed the relation between complex spikes and learning and simple spikes. It didn't lead to a lot of more detailed understanding of the really location of learning, but more as a demonstration of how the prediction of the hypotheses are met. So this is an experiment where a monkey is holding a handle. At the beginning, he needs to move it. I know this kind of movement, and he after a go signal, so we get some kind of go signal. He takes this handle and he moves it, and that's what you see here. So this is just the description of the movement. And then they went, Gilbert and Touch, and recorded in the Purkinje cell. So they recorded both complex spike and simple spike. So in the beginning, you see here, this is what happened before this is just the baseline. So we have this Purkinje cell. They have these simple spikes. When the monkey is not doing much, they have some kind of relatively high baseline rate. And then during the movement, we see this increase in the number of simple spikes. And then when the movement ends, we see this return to baseline. So this is basically coding of the movement itself. And sometimes we see these complex spikes not locked to the perturbation. So this is a highly trained animal that can do this easily, and this is what the cerebellum does. So they record both simple and complex spike. The simple spikes are locked to the actual behavior. The complex spike happen just randomly once a second, with no relation to the actual perturbation that they did in the behavior, with no relation to the actual behavior, just randomly happening.
Where do we see each one? Where the simple and the...
These are simple. The lines here are simple, and this is the complex. Okay.
So there's just one complex?
In this case, there's one. Yeah, it fires at 1 hertz. I don't know what this could be 200 or 300 milliseconds, so you don't have a complex spike at each trial. About 1 hertz. Seem to be random, not related to the details of the movement. And then what they did is change the load of the movement. They add some kind of load to a load that will lead to a learning, or the monkey is getting this load on the handle that he needs to move. And in the beginning, the behavior is very, very different. Now we have this, in this case, it's overshoot, but it's not nice and smooth as you see here. They see some overshoot in the behavior, and then it needs to correct. So the monkey is now introduced to a context where he's not expecting this load. And then what they see is that the complex spike happened exactly at the time when the monkey is sensing this unexpected load. So they're moving their hand, they do this incorrect movement, and exactly at that point you see these complex spikes. Now you see them here. Okay? So then the monkey learns to adapt to that, as they learn and they return after adaptation. They return to some baseline behavior. They return to behavior that's very similar to the behavior in the beginning, but now with load. And then after this, and two things happen here. First, at the time of the complex spike, there was some kind of firing rate of the Purkinje cell. Now the firing rate is lower. So if in the beginning we had here, 7 spikes or 8 spikes, now we have 4 or 5. So that's the first thing. And this is consistent with the idea that these climbing fibers were the teaching signal in order to reduce the connectivity, and then that will lead to less spikes because the connectivity was reduced. And the second thing that you see here is there are no more complex spikes anymore. Right? After he learned the new context, and now we don't have the complex spike. So after adaptation, we're returning to the same situation as here in terms of behavior and a complex spike, but now the amount of a simple spike has reduced.
I don't understand. You said that when you have the first thing that you see is that when you have complex spikes, the firing rate is reduced.
So no, after learning, the firing rate is reduced. So look here, you have here 8 or 7 of these. After they learn, there's a reduction.
The first thing that you see is there's less baseline firing rate and
no, less firing rate related to the behavior. Right? Baseline is more or less the same when nothing is happening.
the beginning or
Compared to the beginning, yeah, and compared to here,
okay, and no more complex spikes.
and no more complex spikes. We don't need that. There's no more error. The animal already adapted.
Here the complex spikes.
So the complex spikes are these dots. This is schematics. You have your.
The dots there.
That's sorry, that's not no more complex spike. No more complex spikes that are locked to the behavior. They still happen spontaneously about 1 hertz, at 1 hertz once a second. And second, just to say that, complex spikes are extremely important for the regular. The random complex spikes are extremely important for the regular coding of the cell. If you take a Purkinje cell and you cut the complex spike, you don't cut the climbing fiber, the cell will completely become regular and won't code anything. So we need this baseline complex spike in order to maintain the properties of the cell. But what's important here is, after we had this complex spike during the learning, the number of spikes after the learning, the simple spike was reduced, and there are no more complex spikes during the time of the movement. Yes?
I'm really confused because we talked about the complex spikes in relation to LTP.
Yeah.
This is like LTP-based learning.
Yeah.
But is there no LTP as well?
So there is an LTP, but the mechanism of LTP are different than the complex spike. I'll just mention them. We know much less about them. But we need at the same way we need an LTD, we also need a different situation than LTD. So this was the first. A more detailed understanding came from the vestibular ocular reflex. I'm not sure. What do you prefer, vestibular ocular reflex or eye blink conditioning? So we'll have eye blink conditioning in the talks, right? So I'll talk now about vestibular ocular reflex, and maybe I'll leave the eye blink conditioning for the talk of the students. So vestibular ocular reflex, I think I showed this. So this is a reflex that we do in order to keep our eyes straight while we move our head. The example is that you put your finger and you move your head from side to side. The eyes are continue to look ahead. Or example, when you run around and you run outside, all the time you move your head, you still want to focus your eyes in the same location. So here's a movie. I think I showed this, right? Yeah, show it again. So in this case, this is a person that will move him up and down, and as a result, the eye will move in the opposite direction. When he's moving up, the eyes are moving down, and vice versa. So, and here is again the canonical diagram of the system. But now we have instead of just talking about inputs and outputs, we have actual anatomical structures. So the input comes from the vestibular. The input to the cerebellum comes from the vestibular organ, goes to the mossy fibers, goes to the parallel fibers, as we talked before, and then the cerebellum itself projects down to the vestibular nucleus. Okay, so this is the same diagram that I showed before, but now we have here vestibular input, vestibular nucleus, and then the vestibular nucleus it projects to the oculomotor neurons or the abducens neuron, for example, that drive the behavior. And in addition, we have information from the inferior olive about the visual motion, the retinal slip. What do I mean by retinal slip? So imagine if we're doing this perfectly, and I'm moving my head from side to side, on the object itself will be fixated on the retina, and we'll have no visual information. But now, if I don't do it perfectly, and there's some the eye is moving a little bit relative to what I'm looking at, we'll have this visual motion that's caused by movement of the object on the retina, and that's what I call retinal slip. And in a sense, if we think of this in the context of control, what we're trying to minimize is this retinal slip. We really want the object to be stabilized on the retina. So the retinal slip serves as an error in this context. When you have retinal slip, it appears that we're doing something wrong in the terms of vestibular ocular reflex. So then the question: what does the cerebellum do in this context? So this is our circuitry, and we can break this behavior into two parts. When we move only our eyes, or when we move our eyes, or when we move only our head. And moving only our eyes is simply the experiment. You move your finger from side to side. And when people looked at the cerebellum, when they looked at activity in the cerebellum, they found that cells are strongly modulated by this stimuli. So this is what I call here eye only. The head is still. The eye is moving in a sinusoidal. The eye is moving sinusoidally because it's tracking some kind of target, and we see these cells that modulate very, very strongly when the eye is moving. Okay, so that's one part of it. So this is the firing rate of one cell. Okay, average what? So this is related to this. This is head velocity. Head velocity is 0, and eye position is changing because, in this case, a monkey is tracking a target. This is a Purkinje cell.
So it's the
Simple spikes, yeah.
You see that the eye position correlated with activity of the
Yeah, it's usually more related to velocity, but there's a correlation, and the details here are not really important. But there's a relation between the simple spike modulation and changes in the eye position or velocity or acceleration. That's what I'm showing here. This is just experiment, moving your eye from side to side smoothly. You see these large responses. And then when we move only on the head, and the way we do this experiment is we have a monkey, for example, looking at a target, and now we move the target and the head together. So it goes like this. And we call this a VOR cancellation. He's supposed, unlike the case of the VOR where you're moving your eyes and head in the opposite direction, in this case you're actually trying to cancellate your inputs that you get from VOR. So this is the case. So and then we go again and look at the simple spikes. This is what you see here. Same as before. This is the simple spikes of the Purkinje cell. Now the head is moving. That's what you see here. The eyes are hardly moving. So the point here is that the signal about eye movement and head movement reach the cerebellum. Okay, both of them modulate the activity of the cerebellum. But the question, what will the cerebellum do in the context of VOR? And when you think about this, it's not that clear because there is this direct reflex. See, there's vestibular input. There's this direct from the vestibular to the vestibular nuclei to the output. It's not clear that we need any modulation on this fine grain because there's this direct reflex. And indeed, what they found is that many cells in the cerebellum actually, when we do the vestibular ocular reflex, when we move our head and eyes in opposite direction, actually are not modulated. And this tells us something about what the cerebellum. I think this is a lesson about what the cerebellum does generally. So the way I think about it, the cerebellum does what it needs to do. And we need to understand this in the context of what's downstream to the cerebellum. So here you see, in this case, the downstream structure basically solves the problem of VOR, right? You don't need it, the cerebellum. And in this case, indeed, the cerebellum does not really modulate it simply. But now, if we have tasks where either there's no vestibular input, as this case, now we need the cerebellum to really drive this movement. Or if we have a task where we need to cancel this, so this maybe is clearer. Here we have this strong input, and we need to cancel this input in order to get the right eye movement. Then we have the cerebellum strongly involved. So this is so we know. Yeah. Okay.
There's the downstream. What's the downstream?
The downstream here is this, right? There's a direct pathway from input to output. Okay. On this pathway, there is you can think about a control circuitry that is connected here, right? And could potentially modulate it. Well, if you think about it, if this pathway completely solves the problem, you don't really need anything to be done here, right? And that's indeed the case. But now, if this pathway does not solve it completely, and this could happen because this could happen because the demands of the task are different. For example, there's no vestibular input as we have here, or we want to cancel the vestibular input as we have here. Then the cerebellum comes into play and modulates this synapse. And another condition where things are not as expected is the context of learning. And that's the next step. So we know the cerebellum is important for these kind of behavior, for moving when you move your eyes and head and coordinate between the two. But when everything is fine, it doesn't do much.
Can you explain how this direct pathway solves the VOR?
Yeah, so you have your vestibular input, okay? Let's say there's now the head is moving in this direction, right? Now you activate this. This is activated. Now this is activated because of that. This is excitatory connection. Now this is connected to the other side of the brain, to the let's say the side that's connected to the muscle. This muscle. So you're moving your head here, and this is connected now to this muscle. You want to move your muscle the other direction. So we have this excitatory connection. This is activated, and now your muscle is activated. That's one muscle. There's more circuitry there, but that's the basic circuitry. So that's what simple spike do during VOR. What do the complex spike do? And it turns out they really encode the retinal slip. One way to see this is that, well, usually this is now let's say this is the target position. You're turning the animal back and forth. And now it doesn't do it all the time perfectly. Sometimes the eye doesn't follow exactly the target, and it moves slower, and that's what you see here. Okay, so there's a difference between the target position and the eye position. And now what you can ask is, when did if we look at the time of a complex spike, so a complex spike happened here, was there any retinal slip before that? Was there a difference between the eye position and target position before there was a complex spike? So this is called spike-triggered averaging. Did you learn that? No. So the idea is to you look at all the spikes that you have this behavior. You do it many times. You look at all the conditions when there was a spike, a complex spike, and now you go back in the behavior and ask what is the average behavior, or what is the average difference between the target and position that happened before and after, right? So the way to understand this is that before the complex spike, there was a retinal slip, and that's what you see here. In another way to see that a complex spike are modulated by retinal slip is an experiment where you simply move the target without head movement. So this is an experiment where the researchers just had a target that moved straight this way. The monkey was required to follow this target. So this is the target starts here, and then moves here, and the monkey follow it. At this point, when they do this, there are not many complex spikes. But now what they did a perturbation when the target moved, and then after some time, 250 milliseconds in this case, the target changed direction. That's what you see here. So it moved like this, and then it changed direction. And what they found is this red curve. Just at the time that the target changed direction, they found a very strong increase in the complex spike. Again, so this is because there was a difference between the direction the eye moved and the direction of, there was a difference between the direction when the eye moved and the direction where the target moved, which was the retinal slip, and they found this strong encoding of this error. And after this, I'm not showing this, but in this context, you can also see there's learning. The monkeys learn to track this. So in the beginning, they move in this direction, then the target makes changes and they don't follow it. But then after some time, they start to move towards the direction that the target will move afterwards.
What was the reason I want
So this is the complex spike, and this is I think this is the complex spike when they do the experiment in the other direction. So there are different kinds of different complex spike have different preferred direction. Ignore the blue one currently. Just that there is a lot of complex spike when there is this error. So we conclude that the complex spike signal the retinal slip, which is the relevant error in this condition. So now we want to ask. So we have this is the coding when everything is working as expected. Now what happens when we have errors in this system? And together with the eye blink condition that we probably won't have time to talk, these were the two I think best examples where there was a strong link between the learning and the actual processing in the cerebellum. And the way we do this is now we have monkeys, or we can have also in human, we can have the same experiment, that wear glasses. They have these, they put these glasses which either magnify the world or reduce the visual field of the world or make it smaller. And the idea now is that if we have glasses that magnify the world, the gain of the VOR is not appropriate. We move our head for a given movement of our head. We now need to move our eyes, let's say, twice the amount that we need to move regularly. And indeed, when you do this kind of experiment, what you can see is that the monkeys can adapt to these changes. So we have here, this is days. You do this experiment day after day. You take a monkey, you put the glasses, you keep them still in the glasses. You have to live with these glasses for some time. And then every day you go and you measure the gain of the VOR. How much the eyes move when you move the head in the opposite direction. So when we look here in the beginning, it's 1. That means the reflex is working perfectly for the regular condition. That means that when we move the eye to one direction, when we move the head in one direction, the eye moves exactly the right, the opposite moves in the opposite movement in the exact same speed. So that's 1. And then when we put the glasses, we see that, let's say it's a magnifying glasses, we see that the gain starts to increase across days. So what that means is that the same kind of movement that we did, the same head movement now causes eye movements that are much larger. So this is this condition, and we have learning in the opposite direction that's decreasing the gain. That's glasses that reduce, that make a 0.5 magnification and reduce the visual field. Then we need a reduction in the gain of the VOR. That's what you see here. And then when we stop it, when we now remove the glasses, we see the after effect returning to normal after some time. So, okay? Yeah.
About point is without the glasses?
Where?
The one that's like
this.
The top, like very high up about.
That's just an error telling, yeah, when we put the glasses, time 0.
Okay, so that's
okay.
Before the glasses?
Yeah, that's before, that's 1. Yeah. This is before the glasses. And then they see this learning.
Sorry, so the gain
how well it is following. And the increase means that the eye movement is becoming bigger for the same head movement. And now it turns out that first cerebellum is essential for this learning that I'm not showing, but basically if you go to the cerebellum and now lesion it, you don't get it completely blocks the learning in this paradigm. You can't learn without a cerebellum. And I'm saying this is opposed to the performance of VOR, in which we can lesion the cerebellum but still get the normal performance for VOR, and that's because, as I said at the beginning, we have this direct reflex that solves the problem. But without the cerebellum, we can't learn. And what you can see here is that there's this is, I know, a reduced diagram, but you see here the cerebellum, and you see here it's connected to the vestibular nucleus with this inhibitory neuron. And there are two pathways. There's one pathway that is connected to the cerebellum, and there's another pathway in the vestibular nucleus that's not connected to the cerebellum. So initially, the first question was, well, is this indeed specific to the cerebellum? Is the pathway that's involved, is the pathway that passes with the cerebellum, or maybe other cells could also contribute to learning? So to do that, we need to find these cells. We need to find the cells in the vestibular nucleus that get input from the cerebellum. And the way to do that is to record the activity in the vestibular nucleus. So put here an electrode, implant a stimulation electrode in the cerebellum, or at least this is the way you could do this. Implant a stimulation electrode here. Now you stimulate here. We know the cells here are connected to the cells here with a monosynaptic connection, and we know this connection is inhibitory. And we can find cells like this, which this is the time where we gave the stimulation. Each line here is one trial, one stimulation. Each dot is a complex, a spike. No complex spike. We're talking here now vestibular nucleus. Each line is one trial. Each dot here is one spike. And you can see here this clear, after the stimulation, you see this clear pause in activity, which means that this cell and this pause is in the right latency. It's monosynaptic latency. So this means that this cell, we call this a floccular target neuron or cerebellar target neuron. This is a cell that gets input directly from the cerebellum. And we have other cells in the vestibular nucleus that don't get this input. So you stimulate and you don't see much. And now we can use this to ask, well, is the learning related indeed to the cerebellar, specific to the cerebellar pathway? And indeed what you find is that if you identify these cells, they would change their activity. So when there's an increase in the gain, so we see here this is the head velocity that they did in this experiment, and this is the eye velocity that you get, and you see that there's a increase in the gain because this eye velocity is much larger than this head velocity. So this is a condition when you have a gain increase. And indeed you find many cells in that increase their firing rate, as you can see here. And when you look at the gain decrease, you see the opposite picture. So these are cells that get input from the cerebellum. But other cells that don't get input from the cerebellum, you don't see much difference between them. And just to emphasize, this is a nice figure, and it gives you the impression that this and this are the same cell in different contexts. These are actually it's not that simple. You can't, because it's a very, very slow learning process, you can't record the same cell for a gain increase or decrease. But at the level of the population, when you look at many, many cells, you see that in gain increase, you see many cells like this, and gain decrease, you see many cells like this. Whereas if you look at the distribution of the population for the non-target neurons, you see they don't really change. So this was the first evidence that indeed the learning was quite specific in the cerebellar pathways in addition to the lesion experiment. Now the question they wanted to ask, where is this exactly happening? And we have here the original hypothesis, what's called Mar and Ito model, and this is what I told you until now. How much time do we have? 10 minutes? We're minus 5 minutes. Oh. Okay, let's stop. We'll do it this way. We have another talk at the end of the semester. I'll continue with it then. Okay? I'm thinking if we can change the day of the lectures of the students.
I think we have no problem with
You don't have problem? What? You'll be okay. Okay, so let's switch that. So the last day, that's the 25th, will be, I think 26th or 25th will be your talks. And lesson before that, I'll finish with this. Is that okay?
Rainy day.
No, it's day before the rainy day. Yeah, it's Sunday.
I would ask Leo.
Yeah, ask Leo, and then I'll figure it out with you. Okay.